Now, I think so. Yeah, okay. So, good. Can you hear me? Good morning and welcome, everyone. So today we are here because we will assist to the thesis defense, the act of the defense of the doctoral thesis of Luis Galan Palma, with that thesis entitled Chromatin Link and FKB-independent IKB alpha function in embryonic stem cells pluripotency exit and differentiation. So I will briefly explain the procedure for you. So we will start with a presentation. You have around 45 minutes, and that will be followed with questions by the committee. We kind of decided to have an informal discussion, if you agree with that, so yeah. We will start with.
Regarding the conclusions, could we just not read them, or like, because I've summarized everything in a working model, so I don't know if your thing is...
With me, it's okay. It's okay. Okay, so we will follow with the discussion with the committee. So let's introduce the committee. First, we have Anna Yannick from the Pompeu Fabra University right here, and Thomas Graf from the CRG, and I'm Maria Abad from, till 2 months ago, I was in Valdebrón Institute of Oncology, and now we moved to Altos Lab Institute of Science in Cambridge, UK. And well, the stage is yours, Luis. Looking forward to listen to your presentation.
Very much.
Shall we move?
Good morning, everybody. I'm Luis Galan, and today I'm going to defend my PhD thesis, which is entitled Chromatin Linked and FKB-independent IKB alpha function in ESL pluripotency exit and differentiation. So to better know what is pluripotency, we need to look at the earliest stages of the development when, after the fertilization of the egg by sperm, the zygote is formed. And this zygote, in very few days, is going to suffer different symmetrical divisions that are forming different cell stages. Until at embryonic day 3.5, this structure is going to be polarized and the blastocyst is going to be formed. And this blastocyst is going to have mainly two parts: the outer layer, which will have the capacity to form the embryonic tissues, whereas the cells from the inner cell mass are the ones that are forming the embryo properly. So cells from this inner cell mass are going to further committing to form later on in the extracellular structure the epiblast, here in dark red. And this epiblast is going to differentiate to form the three different germ layers: ectoderm, mesoderm, and endoderm. That terminally are going to form all the different organs and tissue that will constitute the embryo. So in this sense, one of the most interesting structures from the earliest stages of the development is the blastocyst, since it's the first structure in which you can clearly discriminate between the cells that can form the extraembryonic tissues, which are the outer part, the trophoblast, whereas the ability to form the embryo properly resides on the cells within the inner cell mass, or ICM. And this intrinsic capacity or features that allow the cells from the ICM form the different 3 germ layers is known as a pluripotency. And very interestingly, almost 4 decades ago, it was possible to isolate for the first time these kind of cells from the blastocysts of mice and culture them in a stable way, keeping the pluripotency in vitro. These kind of cells, because of their embryonic origins, were named as embryonic stem cells. And from then, they have been a very powerful tool not only to study development in vitro, but also for regenerative medicine purposes, since these cells have been the source for synthesize different cell products that nowadays are even used in clinics. For instance, from these cells have been possible to synthesize or derive endodermal derivatives such as liver or pancreatic cells, but also it's been possible to differentiate muscle cells or blood cells from mesoderm, or even it's been possible to obtain ectodermal structures such as cells from the skin or even neurons. So understanding this pluripotency capacity that all these cells, the cells have is crucial because it would allow us to better manipulate all the different cell lineages. So in this regard, several pathways have been described and demonstrated that are crucial to maintain and acquire the pluripotency state in mouse cells. However, all of them triggers at the end the pluripotent network, which is governed by the transcription factors NANOG, OT4, and SOX2. And this pluripotency network are going to promote the cell renewal of the pluripotency state, and also at the same time, they will inhibit the differentiation. So more in detail, when you, for instance, stimulate mouse pluripotent cells with LIF and BMP4 or serum, you stimulate at the same time JAK-STAT and SMAD pathways, which are going to favor the stabilization of this pluripotency network. So this is going to be translated into the promotion of the cell renewal of the mouse pluripotent cells, and at the same time are going to inhibit the differentiation. But on the other hand, we have other kinds of pathways that act in an opposite way, such as Wnt/beta-Cat pathway and MEK/ER pathway, that are going to, when they are stimulated, they inhibit the pluripotency network, so they are going to promote the differentiation, and they are going to finish or block the cell renewal of the pluripotency state. But also very interestingly, one of the main pathways that had been involved in regulating immunity and also has been dysregulated in many diseases such as cancer is NF-κB pathway. However, its specific implication at the pluripotent stage is something that has not been very deeply studied. So what is NF-κB pathway? NF-κB pathway is a pathway that, as I mentioned, is crucial to regulate properly immunity, and also it's implicated also in different diseases such as cancer or in autoinflammatory disorders. And also it's a pathway that is is highly conserved from insects to mammals. And in a very simple way to explain this pathway, under basal condition, when there's no ligand bound to the receptor, the NF-κB heterodimer that are mainly formed by these two transcription factors, P50 and P65, are sequestered into the cytoplasm because they are bound by the main NF-κB inhibitor, which is IκB alpha. But when different ligands bind to different receptors, this triggers a signaling cascade that is going to end up with activation of the AKK complex, and this AKK complex, mainly through AKK beta, is going to phosphorylate IκB alpha into serine residues, and this double phosphorylation of IcB alpha is going to lead to be very rapidly ubiquitinated and degraded by a proteasome. And this degradation is going to allow now the NF-κB heterodimer being translocated to the nucleus to induce gene transcription. So, as you may guess, one of the most important proteins of this NF-κB pathway is its main inhibitor, IcB alpha. So, so after its phosphorylation, needs to be very rapidly degraded by the proteasome mediated by ubiquitination to, at the end, allow a proper NF-κB activation. So when you deplete IcB alpha in vivo, you generate mice that have a very short lifespan, since they present a very high NF-κB overactivation, which is then translated into a among other different things, a very severe dermatitis. Since you can see here, the mouse that are like in kappa B alpha present a huge infiltration of immune cells such as neutrophils in the skin. However, also, apart from being ubiquitinated after the phosphorylation of the protein, I kappa B alpha can be also sumoylated. And the sumoylation occurs in the same residue as the ubiquitination. And this sumoylation blocks I kappa B alpha to be, so blocks its protosomal degradation. So this form of phosphosuma I kappa B alpha has been described by our lab to have very crucial and important function at different somatic stem cells. Since it was demonstrated that this phosphosuma I kappa B alpha can migrate into the nucleus of different adult stem cells, and as you can see here, this was something that was described 10 years ago, initially in the skin. You can see that I kappa B alpha was accumulating into the nucleus of at the basal layer of the skin, which is the niche where different skin stem cells are. And also more recently, a similar distribution has been described for I kappa B alpha in other tissues, such as intestine, where this I kappa B alpha is accumulating in the nucleus of the crypts that are where all the intestinal stem cells are. And in a more recent manuscript by the lab, the phosphate kappa B alpha was accumulated into the nucleus of the intra-aortic hematopoietic clusters, which are the origin of the first definitive hematopoietic stem cells. But what is I kappa B alpha doing in the nucleus of those somatic stem cells? So it was again demonstrated by our lab that I kappa B alpha was bound, was nuclear because it was binding to different a specific target, a specific subset of genes that were mainly developmental regulators, such as different HOX and Iroquois genes. And in a basal situation, it was bound I kappa B alpha to inhibit their expression. And also, it was also curious to know that all these genes also were bound by different polycomb proteins that were decorating those genes with the lysine 27 trimethylation. And upon different stimulus such as differentiation or cytokine stimulus, this I kappa B alpha will be released from the chromatin to allow a proper activation of those target genes. So this dynamic binding of I kappa B alpha was allowing a proper differentiation of different adult stem cells. Because when you deplete in those stem cells I kappa B alpha, at basal level, that specific subset of genes present a higher expression. However, upon differentiation clues, that subset of genes cannot be properly activated and the differentiation is blocked. So this fact, the fact that I kappa B alpha is regulating the differentiation potential of different adult stem cells, as well as it was binding different developmental regulators, this raises the question whether this nuclear function of I kappa B alpha could be important even before in the development. So I hypothesized whether this nuclear I kappa B alpha was also required at the pluripotency state. So this was my initial hypothesis. So I hypothesized whether this nuclear function of I kappa B alpha could be mediating the successful pluripotency state to mediate the differentiation of the cell of the first lineages, endoderm, mesoderm, and ectoderm. But as also you know, I kappa B alpha has been described mainly as the main NF kappa B inhibitor. So I was curious to know what was known about this NF kappa B at the pluripotency state. So different works have demonstrated that there is an inverse correlation between NF kappa B pathway and the pluripotency, which is translated with an activation of the NF kappa B upon differentiation of mouse hair cells. And more in particular, seminal work carried out by Fiona Watt's laboratory demonstrated that one of the main pluripotency elements, components that are NANOC, was able to directly inhibit different NF kappa B transcription factors. And in the other way around, when they overexpress all these different NF kappa B transcription factors, such as REL-A, REL-B, or REL, it was enough to directly inhibit the pluripotency genes and also cause a loss in cell renewal and pluripotency. So with this, however, all these, the interplay between these two pathways, NF kappa B and pluripotency, has not been further studied. So with this, I established the different objectives of my PhD thesis, which are first to determine the role of nuclear kappa B alpha in mouse pluripotency and also in the reprogramming towards pluripotency. Second, to study the specific mechanisms by which I kappa B alpha regulates mouse pluripotency and differentiation. And the third one discriminates between NF kappa B and chromatin-related I kappa B alpha functions in pluripotency. However, because of a matter of time regarding the first objective, I will talk only about the nuclear function of I kappa B alpha at the pluripotency, and if you consider later, we can discuss about its putative role in the reprogramming. So I started to characterize how was I kappa B alpha distributed in the mouse cells. So I did some co-staining for I kappa B alpha and the pluripotency marker OD4. And as you can see here in the colony, in the mouse cell colony, I kappa B alpha was distributed everywhere into the cell. However, when we look at higher magnification, also we saw here an accumulation of I kappa B alpha into the nucleus of those cells. So to further confirm this, I isolated protein extract from cytoplasm, nucleus, and chromatin of the mouse cells, and I checked how was I kappa B alpha distributed. And as you can see here, I found very nicely high accumulation of I kappa B alpha into the chromatin fraction of the mouse cells. That was not that evident when we compare the chromatin fraction of other more differentiated cell types such as 3T3. But also, we were curious to know whether how was I kappa B alpha regarding its phosphorylated form. And as you can remember from the introduction, the phosphorylated form of I kappa B alpha is very unstable because it's very rapidly degraded by the protosome. However, and very surprisingly, when we stain for phospho kappa B alpha into the mouse cells, we saw that they were totally positive for this phosphorylated form of the protein. As you can see here also, it was totally coinciding with the pluripotency marker NANOC. So this suggests that the I kappa B alpha that is found into the chromatin fraction is phosphorylated and is quite specific of the pluripotency of the mouse cells. But also I was curious to know whether this was how was this phosphorylated form of I kappa B alpha behaving under differentiation conditions. So I took advantage of this embry-body differentiation approach, which is basically you have, which consists basically in here you have your mouse cells that are growing in a serum that promotes the pluripotency state, which is basically serum leaf. And if you take the cells and seed them in single cells in a low attachment plate in a medium that only contains serum without leaf, this induces a self-aggregation of the different structures, of the different ESLs, that are forming these embry-body structures from which the ESLs are going to differentiate towards the three different germ layers, ectoderm, mesoderm, and endoderm. And after 9 days of differentiation, you can see that the embry-bodies are well differentiated, since just you can see by eye that they are forming beetle-like structure, suggesting that also at that time the number of pluripotent cells that remain are very few. So I took advantage of the differentiation and I stained for some pluripotency marker NANOC and phospho kappa B alpha at that stage. And as you can see here in Davis, very few and only a small cluster of pluripotent cells were remaining NANOC positive, but very interestingly, the NANOC positive cells were the ones that were having phospho kappa B alpha into the nucleus. So this confirmed that the nuclear localization of phospho kappa B alpha was specific of the pluripotency state at the protein level. But also we wanted to know whether this was also happening at the RNA level. So we analyzed RNA-seq from mouse cells and also at embryo bodies 48 hours and 96 hours. And we checked how was the expression pattern of the kappa B alpha gene that was NFK-BIA. And as you can see here, the gene was presenting a similar expression pattern as other pluripotency genes such as NANOC, RES1, PRGN14, CALF4, or SOX2, with highest expression at the pluripotency state, and upon differentiation you can see clearly that the gene is downregulated. And also very interestingly and nice to see was that when we checked other NF-kappa B members and other A-kappa Bs and transcription factors, most of them were showing a totally opposite expression pattern, since they start to become upregulated upon differentiation and clustering together with other differentiation genes such as HOXA2, IOMS, BRCA, etc. So this suggests that, or demonstrate that at both RNA and protein level, the expression of IKB alpha is quite specific at the pluripotency state. So we were curious to know whether this was also having any functional implications. So we decided to knock out the IKB alpha protein by CRISPR-Cas9. So I target at the same time 2 different regions with 2 different guides of the first exon of the gene. And I isolated 3 independent wild-type clones, knockout clones at the mouse CSS cells for IKB alpha. And as you can see, I started to characterize those clones. And when I checked by flow cytometry the pluripotency marker SCCA1, you can see that I didn't see any change in the percentage of SCCA1 positive cells. But very curiously, when we further checked at the RNA-seq level, how was the expression of these other pluripotency markers such as NANOC, GSRRB, SOX2, OT4, CAL4, and REX1, you can see here that the knockout mouse CSL were presenting a higher expression level of those pluripotency markers. But also we were curious to know whether the lack of IKB alpha in the mouse cells was having any implication over the NF-κB pathway behavior. So we took advantage of this KEK database to extract the NF-κB gene signature, and we compare how was this signature between wild-type mouse cells and knockout ESLs for IKB alpha. And as you can see here in the mouse cell state, the behavior of the gene signature was almost the same. And also, more interestingly, upon differentiation of the first stage of the embryo bodies, the behavior of was also the same between both genotypes, demonstrating that the lack of IKB alpha, at least in this early stage of differentiation and the pluripotency, this lack of IKB alpha is not affecting at all the behavior of the NF-κB pathway. But also we were curious to know whether this also was having any implication over differentiation conditions. So I took mouse CSS wild-type and knockout for IKB alpha, and I took taking advantage again of the embryo body differentiation. I differentiated for 9 days, and after 9 days of differentiation, I stained for the pluripotency markers, NANOC and OT4. And whereas in a wild-type embryo body, you can see that after 9 days, very few and small patches of pluripotent cells were remaining. You can see here, when we deplete IKB alpha at the pluripotent state, these knockout embryo bodies were not able to properly silence the pluripotencing program, since it's totally positive compared to a wild-type EB. But also, we wanted to further confirm this increase of the pluripotency cells, so I did some alkaline phosphatase staining, because alkaline phosphatase stain specifically pluripotent cells. So I took embryo bodies after 9 days of differentiation. I disgregated into single cells, and I see those single cells in a medium that in cell leaf that only promotes specifically the expansion of the pluripotent cells. And I did this culture for 4 days. And after 4 days, I just screened for AP positive colonies. And as you can see here in the image, and also here by quantification, whereas in a wild-type condition, in a wild-type situation, the 3 independent clones were presenting a very low number of pluripotent cells remaining. In the knockout cells, you can see that the number of AP positive colonies is much higher. So this confirmed that the number of pluripotent cells that remain after depleting IKB alpha is much higher in this in vitro differentiation. But also we wanted to know whether this was what was happening at the transcriptomic level. So we did RNA-seq of mouse cells and also embryo bodies at 48 hours and 86 hours for both genotypes, IKB alpha wild-type and knockout. And as you can see here, that whereas in a wild-type situation, the wild-type ESLs are able to properly differentiate and exit from pluripotency, the knockout cells, they are stuck in the differentiation and they cannot properly move forward. And more in detail, when we check the pluripotency signature, you can see here that very nicely the wild-type cells are able to then regulate successfully the pluripotency program. However, the knockout cells are not able to do it. And in the other way around, when we check the differentiation programs, only the embryo body wild-types at 96 hours were able to successfully upregulate both mesendoderm and ectoderm differentiation programs. So this confirmed also at the transcriptomic level that cells that are lacking IKB alpha cannot properly exit from pluripotency, and this is translated with a failure of differentiation. But this was a more in vitro system, so we were also curious to know whether this was also happening in a more in vivo and unbiased situation. So we established a collaboration with Dr. Alberto Villanueva's group in EDBale, and we did some teratomas assays. So we took mouse cells wild type and knockout for IKB alpha, and we injected into immunocompromised mice. And we then monitored for 6 weeks the growth of these teratomas. And after those 6 weeks, the pathologists did some histological analysis. And as you can see here, in the upper panel, we have the knockout teratomas from, the wild-type teratomas from 3 independent clones, and in the bottom panel, the knockout teratomas from another 3 independent clones. And as you can see here, the knockout teratomas were presenting higher number of immature structures that are here highlighted by asterisk mark. But also we wanted to know whether this higher presence of immature structures were because of an impediment of silencing the pluripotency in this approach. So we took, again, these teratomas, and we did immunostaining for the pluripotency marker OT4. And as you can see here, the number of OT4 positive cells in the knockout teratomas was much higher compared to the wild-type teratomas. So this also confirmed that in a more unbiased and in a more in vivo situation of differentiation, the knockout cells cannot properly differentiate because they cannot silence the pluripotency program. But here the question is why, which is the mechanism that is causing this blockage in the pluripotency. So there are different epigenetic mechanisms that have been described that are crucial for regulating a proper differentiation. One of the main mechanisms is the one that is controlled by polycomb complex, and this mechanism, what is causing at the pluripotency state is keeping the chromatin in a permissive state. So this allows, and upon differentiation, this allows proper activation of all the differentiation programs and a successful pluripotency exit. But on the other hand, other main mechanism that is involved in a successful differentiation is the formation of the heterochromatin mediated by lysine 9 trimethylation. Because upon differentiation, this lysine 9 trimethylation is deposited in different pluripotency genes that at the end are going to cause a very stable silencing, because it's going to mediate the methylation at the DNA level of the promoters of the pluripotency genes. So these are going to mediate a very stable silencing to ensure a proper differentiation, a proper activation of the differentiation. But also very interestingly, this mechanism is also important already at the pluripotency state, because the lysine trimethylation is also inhibiting different retrotransposon elements and also satellite repeats and also imprinted genes already at the pluripotency state. So we decided to study both epigenetic mechanisms, and we started to characterize whether there was any change in polycomb function. So what we did was to take and did chip-seq of mouse cells, wild type and knockout of IKB alpha of the lysine 27 trimethylation, which is the mark deposited by polycomb. And as you can see here, the mouse cells lacking IKB alpha was having a change in the distribution of the mark, since there were some regions that were gaining presenting higher levels of lysine 27, and other regions that were having lower levels. More in particular, the genes that were presenting higher expression of lysine 27 trimethyl were genes that were belonging to a general processes such as regulation of ion transmembrane, regulation of transporter activity, or even hormones transport. But on the other hand, the genes that were presenting lower level of lysine 27 trimethylation were genes belonging to a very specific developmental processes such as pattern specification, embryonic organ morphogenesis, or even mesoderm and mesendoderm differentiation. So is this change in the distribution of lysine 27 having any functional implication? So what I did was to take the genes that were having differential lysine 27 binding and it checked how was their expression at the mouse ear cells. And as you can see here, although I saw some genes that were presenting different expression patterns, I didn't observe a huge dispersion of the expression of those genes at the mouse ear cells state. However, upon differentiation, the same genes that were presenting differential binding of lysine 27 trimethylation, they were genes that upon differentiation at 96 hours, they were mainly downregulated. So these results suggest that this differential distribution is causing a failure in the activation of mainly differentiation programs. However, I attributed to a more mechanism related with differentiation, and as you can see from the previous slide, the changes in the distribution of lysine 27 was not huge. So this was not correlating with that severe phenotype that we were seeing regarding the pluripotency blockage. So we decided to explore other epigenetic mechanisms, which is one of the key that one of the key mechanisms that regulate cell identity and very stable silencing, which is DNA methylation. So I started to characterize how was the DNA methylation level in the mouse ear cells. So I did immune staining for 5-methylcytosine, 5-MC, which is the mark that is related to DNA methylation in the eukaryotic cells. And as you can see here, the mouse ear cells wild type were presenting clearly regions of that were methylated at the DNA level. However, upon ecopapillary alpha depletion, you can see that the number of methylated regions at the DNA level was much reduced. So to further explore what was happening there, we established a collaboration with Dr. Manuel Esteller's group in Josep Carreras Institute, and we did some DNA methylation arrays. And as you can see here, we did the methylation arrays in the mouse ear cells. And in the wild-type situation, you see here some regions, some genomic regions that were not having any kind of DNA methylation, and other regions that were presenting methylation at the DNA. However, upon icapavir alpha depletion, the number of genomic regions that were losing totally any kind of DNA methylation was much higher. And also, the regions that were presenting some degree of DNA methylation was, compared to a wild-type, also present lower levels of DNA methylation. Moreover, we did the same analysis at the embryo bodies at 96 hours, and you can see here that in a wild-type situation, there's a clearly increase of DNA methylation, which is correlated with a successful differentiation in a wild-type situation. However, the knockout embryo bodies, although they are also gaining some kind of DNA methylation, the levels of DNA methylation are much lower compared to the wild-type. So this clearly demonstrated that the DNA methylation was at least until at some degree abolished in the knockout cells. But we wanted to further explore all these mechanisms, so we took the genomic regions that were hypomethylated in the mouse ear cell knockout, and we crossed those regions with the regions that were hypomethylated in the embryo body's knockout at 96 hours. And as you can see here, we identified different regions, and in particular, we identified these genomic regions that were specifically hypomethylated in the mouse ear cells knockout, and you can see that they were genomic regions in which different pluripotency genes were contained. So this demonstrated that the pluripotency genes were not successfully being methylated at the DNA level upon differentiation occurs. But also, and even more interestingly, we identified this region that was common in terms of hypomethylation between both time points in the knockout mouse ear cells and into the knockout EVs. And when we further explore these regions, you can see here that they were totally hypomethylated in terms of DNA methylation, and within these regions, we were finding some genes such as PLAGL1, PER13, MES89, or GRB10. But even more interestingly, when we did some functional analysis, of these regions that were commonly hypomethylated, we saw that these regions were typically decorated with the lysine 9 trimethylation mark. And even more interestingly, we saw that also these regions were bound by the ZDB1 trim28 complex, which is the complex that deposits the lysine 9 trimethylation mark. So this data suggests that upon ecopapillary alpha depletion could be some affectation of this lysine 9 trimethylation deposition, which at the end are going to lead with a failure of the DNA methylation. So to test this hypothesis, or to tackle this question, we did some ChIP-seq of lysine 9 methylation in the mouse cells, wild type and knockout for AKPB alpha, and as you can see here, very clearly, there's a clear reduction of the mark of lysine 9 methylation mark in the knockout mouse cells. So here also you can see even more specifically in the genome browser for these 3 genes that were also genes that were clearly hypomethylated. At the DNA level, you can see that these genes in a normal situation in a wild-type mouse cells, they are typically decorated by lysine 9 methylation. However, upon AKPB alpha depletion, there is a totally global loss of this mark, of these histone marks, which is very nicely correlated with the gain of the lysine 43 methylation, which is a mark that is linked to a totally different process, which is gene activation. So also I was curious to continue studying this mechanism, and I did some immunostaining of the mark of the lysine 9 methylation mark in the mouse cells, and as you can see here in the wild-type mouse cells, the mark is very well confined to specific regions. That this is something expected because, as I mentioned before, this is a mark that is at the pluripotency state is found specifically in highly repetitive elements, in retrotransposons elements, and also in imprinted genes. However, upon AKPB alpha depletion, the distribution of the mark is totally different. But very interestingly, this change in the distribution of the lysine 9 methylation was not related with a global change of lysine 9 methylation. Since you can see here between wild type and knockout cells, we didn't see any change in the global levels of lysine 9 methylation. But also I was curious to know whether this effect over the lysine 9 methylation could be because of a direct effect of AKPB alpha. So I was wondering whether this AKPB alpha could be interacting directly with the main lysine 9 methyltransferase, which is ZDB1. So we did IPs of ZDB1 to address whether it could be interacting with AKPB alpha. And for that, I took advantage of a mouse ESL line that were overexpressing just a mouse ESL line which was background knockout for AKPB alpha. I introduced an overexpression cassette of AKPB alpha to regulate by doxycycline, and as you can see here, after expressing AKPB alpha, was able to interact directly with ZDB1. So this could explain that the effect of this lysine 9 methylation deposition and also DNA methylation was a direct effect of AKPB alpha. But also, as you remember from the introduction, typically AKPB alpha has been the main inhibitor of the NF-κB pathway, but also our lab has described a novel and alternative function which is related with a nuclear distribution and interaction with histone. So far, nobody has been able to discriminate between these two functions of AKPB alpha. However, because of terrific work from Dr. Jose Espinosa's laboratory, it was possible to generate separation of function of AKPB alpha for only NF-κB function. So we were able to generate mutants for AKPB alpha that only can bind NF-κB and cannot bind any kind of histones, which is here AKPB alpha delta chromatin. And on the other hand, we were able to generate other mutants for AKPB alpha that were not able to interact with NF-κB, mainly the heterodimer P50, P65, was not able to interact with this complex. However, it was remaining the ability to interact with the histones. So you can see here, whereas in a wild-type situation, AKPB alpha can interact with the P50 heterodimer of NF-κB, can interact, and also can interact with the histone through the histone H2A. We were able to, in the case of the AKPB alpha delta NF-κB, it was not having the ability to interact with NF-κB, but was remaining the ability to interact with the histones. And on the other hand, the AKPB alpha delta chromatin was not able to interact with the histones, but it was able to interact with P50, so with NF-κB. So taking advantage of these different mutants, I generated different cell lines that were background, and they were having the background knockout background for I kappa B alpha, and I generate different cell lines to overexpress by doxycycline. The wild type form the I kappa B alpha that cannot bind NF kappa B, and also the I kappa B alpha that cannot bind the histones. And I use them to try to reconstitute the experiments. And I initially was curious to know whether just by overexpressing the different I kappa B alpha forms, it was enough to rescue the DNA methylation level. So what I did was to take the mouse cells and start giving doxycycline for different passages. And at the third passage, I did immunostaining for 5-methylcytosine. And as you can see here, it was able to, the overexpression of I kappa B alpha, wild type, and the one that cannot bind NF kappa B was enough to increase again the levels of DNA methylation at the mouse cells. However, when we compare how was the level of the cells that were overexpressing the I kappa B alpha that cannot bind the histones, you can see that they are much reduced, demonstrating that this restoration that the DNA methylation was specific of the chromatin function of I kappa B alpha. But even more interestingly, I was interested to know whether this overexpression of I kappa B alpha was enough to rescue the differentiation potential of the cells. So I took the mouse cells and I overexpressed I kappa B alpha upon embry-body differentiation. And as you can see here, I was able to overexpress I kappa B alpha more or less at the different I kappa Bs at the different, more or less at the same level. However, and very interestingly, when we immunostain embryo bodies at 9 days of differentiation, you can see that only the ones that were overexpressed I kappa B alpha wild type or the I kappa B alpha that cannot bind NF kappa B was the ones that were restoring the differentiation potential. Whereas on the other hand, when the EVs that were having an overexpression of I kappa B alpha that cannot bind the chromatin, only being able to inhibit the NF kappa B, the number of pluripotent cells that were remaining was almost the same as a knockout. Also, we confirmed this at the bialkaline phosphatase. So you can see here that wild type cells are having very few cells, AP positive cells, that are much increased in the knockout situation. However, when you overexpress I kappa B alpha wild type and I kappa B alpha that cannot bind NF kappa B, only the histones, the number of AP positive colonies are much reduced, suggesting that, confirming again that the number of pluripotent cells are much lower. However, when we overexpress only the I kappa B alpha that cannot bind the chromatin, that only remains the NF kappa B inhibition, the number of AP positive colonies are not reduced. So with all this previous data, we have built this working model. I think that we can discriminate between two subsets of genes that are regulated by I kappa B alpha. One subset of genes, I think, is that are regulated by I kappa B alpha are also related with a polycomb deposition. And I think also these are genes that are more important for a differentiation state and also for activity in the differentiation program. And whereas already at the pluripotency state, I think these kind of genes are not that important. However, we have another subset of genes that are the ones in which I kappa B alpha could be mediating the deposition of the lysine 9, mainly through its interaction with ZDB1. And this deposition of lysine 9 trimethylation, I think, is crucial to, at the end, establish silence at the DNA level, the genes that need to be silenced to, at the end, ensure a proper pluripotency fate. So these are the conclusion of my PhD thesis, which have been summarized in the working model. So that's all. I would like to thank everybody, all people in my lab and all the family and everybody that have been supporting me during this year, all these years and this pathway. And also would like to thank everybody for the attention.
Thank you very much, Luis. So before we start with the discussion and the questions by the committee, we should ask is there's anyone in the room with a PhD degree that wants to ask something, including the director of have any other comment? Apparently not. Okay. So we will proceed then with the discussion. As we said before, I think we all agree that it would be most rich informal discussion with comments for every member of the committee when appropriate. We can start with Anna, and then if we agree, Thomas, we can interrupt at any moment. Yeah, that's good. So Anna.
So first of all, I would like to, do you hear me? Yeah.
Yeah.
So I would like to congratulate you for your wonderful thesis, and I really enjoyed reading your thesis, but I can say that I much more enjoyed your talk as well, because it was very neat and very, very easy to follow. So congratulations to you and to your supervisor on a beautiful work, because I think combining, you know, these omics data with, first you get great phenotypes and then omics data, and then after that you actually molecularly dissected what is going behind this. So I think it's really, really nice. So congratulations. And so I want to say that, you know, the schemes that you have is just global are very nice, and the figures also. Maybe just a few things, you know, that I want to mention. Maybe, you know, the references. Sometimes I'm missing, you know, them maybe through the discussion as well, you know, because there were some statements and also maybe a little bit more updated, but this is just the minor thing, you know, just to say something. Anyway, so then I start with the questions. So I was thinking, so first that came on my mind with your presentation and also reading your thesis is that this AKK alpha is so important for the stem cell functions, you know, in the mouse. And then this mouse actually lives, you know. so I was actually, yeah, because it forms all the tissues, no, eventually. Yeah. So I was just thinking, what is your reflection on this? Because for me, it was remarkable.
Yeah, this is something that I've been asking myself since the first day when I started to observe this phenotype. I don't have any certain answer, just totally speculation. I think one possibility is that in vivo there's some kind of compensation, at least at the gastrulation level, when the first stem cells, the pluripotent stem cells are. But after once they form the gastrulation, I mean, we've been checking in the embryo once the tissues are formed, and all of them are having problems of cell identity and proper differentiation. But I think that regarding at the pluripotency state, I think it has to be some kind of compensation. As I don't know, there are some, for instance, some polycomb subunits that then in vitro are having phenotype, and then when you check in vivo, they are not having the same phenotype or that severe phenotype. And also another possibility that I was thinking is that here we are maintaining in vitro an state is very artificial, because pluripotency in vivo in the mouse is like 2 or 3 days maximum, no? Once the gastrulation is lost. So one possibility also is that we remove the protein, the A-kappa B alpha, and from the day in which we knock out the protein and until the day in which we start the experiment the days have been running 2 or 3 weeks. In the meantime, we're isolating the clones. So maybe another possibility is that we are seeing that clear phenotype because of a matter of time, because if it's a passive loss of DNA methylation, for instance, this is something that is amplified upon passages, as it has been demonstrated with other enzymes that are or proteins involved in DNA methylation. So, yeah, I think, and yeah, but of course, I think that nobody has checked before in vivo, for instance, in the blastocyst. So I think this is something that we need definitively to check to see how is the lysine entry methylation in vivo, also DNA methylation, because I think nobody has checked before.
Yeah,
so.
Very, very interesting, yes. And, yeah, go.
No, I have a related question. If you look at early embryo development, what is the earliest stage at which you see I kappa B expression, and is it restricted to the ICM?
In vivo data of blastocysts I have not checked, like single cell or in which they discriminate. I just checked the mouse cells and also at the earliest stages of 2 cell, 4 cell. And very curiously, I found a paper that was published like last year or 2 years ago, in which they start checking the zygotic genomic activation in human. And very curiously, they were speaking about very few genes that were the ones activated at that stage. One of the few was I kappa B alpha. So, and I've been checking also that I kappa B alpha, at least in human, can interact with DUX4, which is one of the key proteins that mediate the zygotic genome activation. So I guess that it has to be important also as early as even at the zygote, but honestly I've not checked yet, specifically maybe single cell from the blast or to know whether it's specifically checked in the trophoblast or in the ACM. This is something that I still need to look.
But in the blastocysts, have you?
In the blastocyst, I checked RNA-seq, and that is in one of the figures,
Yeah.
and it's expressed. When I check the different A kappa Bs,
But what about ICM versus trophectoderm?
I'm not checked.
Okay.
This is something that I need to look definitively because I'm very curious to know if it's higher or at the same level expressed.
In this regard, it would be really interesting to just microinject the CSL that you have.
Maria.
Oh, sorry.
Utiliza.
Sorry for this. Yeah, it would be really interesting to microinject the ESL's knockouts in blastocysts and see the outcome and see the different patterns of how they are behaving, no? Or compensating in vivo.
I have to say that the other day I was reading and I found one of the papers from Konrad Hochadlinga in which they were, when they put the mouse cells in a very long term into a leaf that stabilized the gram state, the nice pluripotency, then they started to observe a very similar phenotype because they are the cells were losing DNA methylation in imprinting regions. They cannot properly differentiate, and also when they injected, they were showing much lower contribution of the cells. So this is something that is very interesting, and maybe it's a great experiment to perform.
Or maybe if there is a competition, no? I guess as well, because you have a beautiful mutant, no? That you have a wild type and a mutant, and maybe a competition assay in vivo, injecting in blastocysts.
Yeah, I think also now that we have the mutants, making the knock-ins, because this is just based on overexpression, and I have to see that the reconstitution experiments are quite tricky, because I think also the expression level of the protein regarding A kappa B alpha is quite important, because if I just use the standard like induction with doxycycline that are making a huge A kappa B alpha overexpression, then the cells cannot differentiate properly, neither. So I think with the knock-ins and performing this kind of experiments with the knock-ins and also even going in vivo with the specific A kappa Bs would be, I think, great.
Yeah, in your teratomas, so the ones that you had, so have you, because you have shown like the histology slides and everything, but have you measured the teratomas? The sizes, are they different?
Just by eye, we didn't see a huge difference. We didn't see that the teratomas were smaller, or we didn't see that difference. I think this is something that we are not experts, so the pathologists is something that they have done. But also just by eye, the size of the teratomas were not different. And it's when they were analyzing the grade of differentiation, when they started to see that they were forming more immature structures. And also, regarding the formation of the 3 different gene layers, at least so far, we still need to, because this is something still ongoing, but we have not seen any bias in terms of more towards endoderm, mesoderm, or ectoderm. It's been more related, I think, with maturation of the differentiation, which makes sense also with the phenotype in vivo of, for instance, in the intestine or in the skin, that also they are much less differentiated. So
What about the incidence of teratomas? You didn't say anything about that.
we didn't see differences.
Would you
They
not
were
have,
forming teratomas in those situations.
but would you not have expected to see that?
You mean translating into a lower teratoma incidence in the knockout? So
Yeah, I mean, what if you compare wild type with the I kappa B defective and you make teratomas? Do you see a difference there?
in the formation.
Yeah, in the frequency of.
Not really, but it's also, we have only made one experiment. So we injected different mice from 3 different clones of each genotype, but the number of mice that we injected was with the number of mice we were injecting, we didn't see a much, at least just think, oh, there's a clear difference between situations. We didn't see any difference or major difference. But maybe something that we should measure, because honestly, this is something that, I mean, it was the first time we were not experts, so probably now, as you know much more, this is something that is worth try and something that we need to measure.
If you repeat the experiment, I agree with Thomas and also with Anna, that it would be really informative, because if you are expecting a defect in the exit of pluripotency or an effective differentiation, you should expect higher sizes, higher proliferation, so there's something that you can also check the proliferation in the teratomas, and also the latency.
Yeah, I think this is something that we have to keep in mind to do it. So
And maybe as well, I don't know, maybe it's too much, but for the next expert, but to rescue as well, to do the rescue experiment. That would be fantastic.
That's, I think, is the experiment, the key. Yeah, exactly. Yeah, thanks.
So I have a question about how do you think, how does I kappa B bind to chromatin? What is the glue?
I think so. It cannot bind directly to the chromatin if it binds through the histones. This is what it has been demonstrated, and what we've been seeing is that it can interact with histones.
Which one? Is it specific?
In other cell types, we've seen that can interact with histone 4 and histone 2A. In mouse cells, we've been able only to see the interaction with H2A, not with histone 4. So I don't know if it's also a matter of how is the chromatin state, how is the histone mark landscape. This, I think, is something that we still need to see, because at the pluripotency state, because what it has been demonstrated with other models, specifically in the skin and intestine, because it's where all the main work has been done, is that it can interact with different lysine modification of histone 4 that are, I've been checking, like for instance, can interact with the lysine 20 trimethylation of histone 4, which is related with repression and heterochromatin. But I think how is mediated at the pluripotency state, I mean, it's something that we still need to characterize. But also,
I mean, wouldn't you expect that it has to be a rather specific interaction, not just all histone 2A or across the board, or histone 4, because otherwise it would be covering all genes, right? But you see very specific effects.
I think also it's worth, of course, we need a cheap seek, because I tried, and the antibodies that are for ICOPAVs are very hard to work for, doing things with the chromatin, because it's not, they are not for that. So now I'm tagged the endogenous gene, or I'm tagging. But I think, regarding your question, Thomas, it has to be not like everywhere. I think it has to be, at least in the mouse cells, in specific regions related with constitutive heterochromatin, because I think the main effect we see at the DNA methylation level are regions of heterochromatin which are the ones that are close with the lysine 9, and I don't think that is something like everywhere, and also because I think it's something quite specific of specific genomic regions, because also this could also explain why we are seeing are seeing like embryo lethality, maybe because it's regulating a specific set of genes. But I think it's more related with the inhibition and keeping I would say keeping the heterochromatin formation in a good way. Because for me...
Yeah, but that would explain the defects in differentiation. It wouldn't explain why it maintains this high pluripotency state. You know, how does it make it, the pluripotency network, so stable?
I think it's dampening the expression level of pluripotency, because it's like acting a kind of brake to maintain an equilibrium between naive because in this serum leaf condition, cells fluctuate, even they are in naive, they fluctuate more. One are more prone to keep the naive, and the other are more prone to differentiate. So I think ICAPAB is keeping that equilibrium, because when you remove, you stabilize the naive program, because I'm not showing here the result, but we've been crossing RNA-seq data from this heterogeneity that are observed in serum leaf, because we took a paper and crossed the gene signature from the different populations, more like prime and more naive, and we saw that our knockout for ICAPAB alpha were enriched in a more naive state. And also when we check the lysine 43 methylation, that this activation, we saw that the main genes that were gaining also lysine 43 methylation were genes related with naive pluripotency. So REX1, CALF2, and among others. So I think it's keeping the right dose of expression of the pluripotency genes that are required, because if you remove, then they just become up, and it creates like a super pluripotency state, as it has been observed with other factors that is crucial to exit from pluripotency.
Here maybe I can jump in. In fact, one of my question is, what if you tested if there's a shift between ground state and naive state or prime state, that you just answered, that it's really, really interesting. And also regarding that, you just said that you checked the H3K4 methylation, because I was wondering, reading your thesis, that you said there's a defect in H3K27, but those genes are bivalent genes or not? I was because they are so involved, no, bivalency of these two marks in the same developmental genes, that would make all the sense that you have defects also or affected in those genes, the H3K4 methylation.
We checked specifically the bivalent genes, and we have not seen a change in the differential. At least, statistically, we have not seen, just running the differential binding of those two marks. They are not different.
So you checked the expression of...
I checked the expression. Some of them are upregulated in the knockout, but then when we check, like for instance, SOC17, that is one of the bivalent genes and related with more endodermal differentiation. Curiously, they are like, in the knockouts are upregulated, but then when we check the epigenetic landscape, lysine 27 trimethyl and lysine 40 methyl, we didn't see changes. So maybe it could be...
But did you perform ChIP-seq of H3K4 methylation?
Yeah.
You did?
Yeah.
It's in your book? Or?
No, in the book, no.
Okay.
No.
I was looking for that.
It's been later, sorry.
Okay.
Yeah.
Okay.
But specifically all the bivalent genes, there's a list that was defined by Presley-Bernstein. We checked that list, and none of the bivalent genes were affected Some of them at the early expression, yeah, we saw that some of them was under basal condition upregulated, others downregulated. But in terms of losing lysine 27 or gaining lysine 4 or some kind of imbalance between these two marks, we didn't see any real change, like to see like a severe change. So
it's it's interesting, and thinking about the publication, I think you can include this data.
I think it's a good suggestion. Thank you very much.
So I have a question related to your protein, this SETDB1. So the methyltransferase that you have found that interacts with AKK alpha, no?
Yeah.
So because now you found, because I haven't seen in your thesis, you found that it's not there, no?
No.
Okay. Because
because
I was asking myself, is it direct or indirect? But now we know it's a direct interaction.
Yeah,
but then my question is more related because then I was reading that this, in your thesis, that this protein actually is leading to the totipotency as well, no? So would you think, if now you have this relationship between them, do you think that maybe your protein as well has a role in the totipotency?
I've been checking markers.
Yes.
And also like of the two-cell stage, like Dukes is one of the main. And I didn't see differences. So I think it's more related with the stabilization of the naive pluripotency, with the ground state pluripotency, more than like a reprogram towards an earlier stage. Because all these markers that are signature of totipotent or two-cell-like stage, I've checked and I've not seen any difference in the expression. So I think it's a more stabilization of the naive pluripotency. So, which also, now that you are seeing this, I saw that there's a screening, a CRISPR screening, which they just investigate the mechanisms that were stabilizing the prime or more naive pluripotency, and they saw that the IKB alpha knockout was enriched in the naive state of pluripotency when cells were cultured into eye. So this suggests and confirm like that it's important to expand or stabilize the naive pluripotency. So
is there an inhibitor of? I was going to ask you that question later on, but now, like, because we are using 2I or this CDK8 inhibitor to stabilize the naive state, do you think you can develop, or is anything an inhibitor to just stabilize this, or to prevent differentiation in vitro? That is the nightmare of every researcher in the stem cell field, right?
So I think one of the main ones are related to inhibiting the phosphorylation of AKB alpha, so inhibitor of the AKK beta, AKK complex through NF-κB. But one of the problems with this, I don't know if there's any, I have to be honest, and I don't know if there's specifically a direct inhibitor of IKB, but the main one says just to block the AKK complex to avoid the phosphorylation of IKB alpha that then leads to a chromosomal degradation. But one of the things, the NF-κB pathway in the mouse cells, there's no almost any NF-κB activity. When you try to inhibit with this kind of inhibitor, so stimulate with TNF-alpha, you didn't see much changes in the behavior of the cells, because I think the expression of the receptors and all the components of the NF-κB pathway are very, very, very low in the mouse cells. So I think, if we can find a specific inhibitor, it would be very nice.
I have a very sort of detailed question. Looking at your results, I had a problem understanding the figure that describes your chip enrichment analysis. And you say you listed top 5 genes, and then there are 3 times SUTS12. Why? I just couldn't understand that. And that's true for some of the other parts of this figure.
I have to say that this is just in silico analysis because
Yeah.
we took the differentially expressed genes in the mouse cells, and we just did this cheap enrichment analysis to see what could be altering the genes and could be regulating the expression. And it's when polycomb, all these genes there, or all the proteins related with polycomb appear. So.
But why 3 times the same
sorry.
gene?
Yeah, because this database takes like different experiments of ChIP6. So this means like with SUS12 in the figure appear 3 times because in 3 independent experiments it's correlating with SUS12. That all the genes that I've been submitting to this database are coinciding with 3 independent experiments of SUS12 ChIP6.
I see. So they're not really the top 5 of one experiment, but there are top 5 of many different data sets.
Like here is the top 5 of the major overlapping with the ChIP6 from SUS12, 3 independent ChIP6 of SUS12, another for SAP4, another for MTF2.
I see. Okay, that makes sense. Thank you.
Thanks, Bill.
So if I can ask another question. You mentioned that maybe a limitation of understanding the immediate early effects of I-kappa-B is that you have generated these clones, given them a long time to develop, giving them time to develop compensatory mechanisms, and you don't know what you're selecting for really. So if you would start again, how would you do that to avoid this problem?
I would use either Protax or some Degron system, I think. Because with the Degron, you control in 15 minutes. With Auxin, you remove the protein, and then even you can revert the expression protein. So actually, at some point else I wanted to, I don't know if I will have time, but do Degrons of I-kappa-B alpha to deplete specifically upon the first stage of pluripotency exit, and then restore the expression to see, to define specifically the window in which this I-kappa-B alpha is required. I think it's much more cleaner because with the knockouts, to remove is okay, it's super clear that you have the knockout, but yeah, then a lot of things can happen, and then you see something like in a long term. So yeah, I would definitively do Degrons.
Yeah. Good.
Maybe I can ask the question. So, because you got some, as far as I understood, some differences that you see, for example, with methylation of the H3K27, no, that you see between the mouse cells and actually the ones that are mouse embryonic stem cells and the ones that are in adult stem cells. So, why is this difference actually happening?
You mean like the differences in the
in the methylation that you have seen, in the global methylation of the H3K27, no?
Like in the mouse cells and in the bees?
Yes.
I think, so in both situations, I just compare the expression level of the genes that were having differential lysine 27.
Mm-hmm.
I didn't do a ChIP6 of the lysine 27 in the ABs. So this is something that maybe we can do. But what I did was to see whether the genes that were having differential lysine 27 already at the mouse cells, how was their expression upon differentiation. And I saw that, that's why I was saying that I think it's important for activating some differentiation programs, because all the genes that were having mainly loss of lysine 27 were genes such as FOXA2, IOMS, WIN3 that are critical for activating the mesoderm commitment. So I think maybe, I guess, since we don't see like huge and crazy changes of the lysine 27 in two genes in the mouse cells, I guess that maybe if we do the same ChIP6 already in the EVs, maybe we will see that strong reduction of the lysine 27 mark. But I think this is something more related with activating the differentiation, more than with pluripotency.
Yeah.
I have a couple of technical questions that maybe I can just now. So you said also during your presentation that it seems that the phosphorylation of AKB is necessary to the nucleus. Is that right?
Yes.
Okay. Because I think that there was the immunofluorescence in the book are not. You show something else, no?
Because these are the phospho alone, and then in the presentation also
you showed the total one. Okay, that was one. And so I was realizing that the pattern, I mean, you have in the Western blot in figure the R1 and R4. So the pattern of there's a different bands, right? And it seems that maybe is just the chromatin fraction, the chromatin AKB that which has several bands pointing to phosphorylation or some others modifications. So have you tested that, if those bands that appear in the chromatin fraction are in fact phosphorylation?
I have to say that specifically in the monocyte cells, it's been very difficult because in the monocyte cells there are several bands of A-kappa-B alpha that you need to compare with a knockout to see which ones are specific. So, and also in particular, I think the ones that are highlighted here, like as a I-kappa-B, that is like in darker black, like in black, are the ones that are disappearing in the knockouts. So, related to the question that if it's modified, in the monocyte cells, when I did the phospho-A-kappa-B alpha, is at the 37 kilodalton size. So, but what it also been demonstrated in other systems in our lab is that when it's humulated, it changes.
Shifts.
It shifts. But I'm not being able to see that form still in the monocyte cells. In the pluripotent cells, still I'm not being able to see.
Maybe with a sumo inhibitor.
I did it.
Okay.
But we are still trying because since it's very unstable, it can be related. We cannot discuss. One thing is that we are trying, not me but Amelis, try to do IPs of sumo and then blood A-kappa-B to see whether it's sumo related. So, but I'm not seeing clearly that this shift in the position of the sumo A-kappa-B, I'm not seeing it in the pluripotency cells.
No, but just comparing the figure 4 with the figure 1.
Because also here, this is also cytoplasm. I just to screen
the total
screen because to be fast, I just was doing Western blot and I just wanted to isolate the knockouts. So
this is only the cytoplasmatic
version.
It's not total lysis.
No, I just did soluble from cytoplasm just to see.
So then it makes sense that it corresponds to the same pattern than it. Okay. And also just another technical question in figure 8. So it seems that, I mean, the phenotype that you have with the embryo bodies is really clear to me, that you're maintaining pluripotency in the embryo body. This is really clear and really robust throughout all the thesis. Especially regarding what Anna pointed out, that there's a controversy between what Joseph with the phenotype of the knockout. No? Maybe it's important to just strengthen the differentiation protocols and techniques that you're performing. On one hand, I was finding some also discrepancies in figure 8, sorry. You see that the knockouts have a defect in mesoderm and endoderm differentiation, but increased ectoderm.
Yeah.
Which is the opposite that you show afterwards, that there's a defect in just the 3 lineages.
Yeah.
So
specifically these 3 genes were behaving like like the opposite, like increasing. But you have to see that when I increased the uncheck, take real signature of ectoderm with a lot of markers, is when then I saw a clear difference that also they cannot differentiate, because in one of the figures when I was here I'm almost there. here. So you see here that when I increase, so this in the figure 8 are just qPCRs, in which I just select some markers that were published in the literature. So, but then when I just took like a real signature of every process, such as pluripotency or then mesoderm, endoderm, and ectoderm, I do see the difference. Actually, it's true that in these genes are, you see this tendency of being
higher.
But then checking others, then I saw that they were totally like the same.
Okay. No, just to avoid confusion, because at the beginning, reading this figure first, I said, oh, maybe there's it's an increased neurectoderm because you have less endoderm and mesoderm. There's a shift, no? But there's no shift. It's just that they stuck in pluripotency, which is different, no? Than the switch. So this maybe can...
Actually, when we saw, because this was one of the first experiments when we were doing the knockouts, and when I saw this, I thought the same, like, oh, they are polarized. So I tried to some ectodermal differentiation for neurons, and then checking markers, I didn't see like differences.
Okay.
So like it was the same, having problems in differentiation.
Okay.
So I'm not included here, but maybe it would be also
it's good to know. Yes. And what regarding to what I was saying, the strengthening, of course, the template bodies experiments are really clear, no? The teratomas, you already said that you are working on improving the characterization of the teratoma. We just discussed about including some other measurements as proliferation, the size, the latency, and the tumor burden as well. But there are some other differentiation protocols. Have you tried retinoid casing differentiation, for example?
I tried with, for the neurons, I tried EVs with retinoic acid, and it's when I was saying also that I saw also a blockage in the neuroectodermal differentiation. With other, for instance, just try to direct specifically towards mesoderm, or I have not tried, I just did EVs. But I think also it would be also interesting to see and derive maybe with FGF and activin to form or to address whether they can form APSs if they are stuck more in pluripotency.
I was just thinking if the knockouts are preventing the retinoid casing differentiation, that it is just kind of a bump. It would be really surprising, no?
I have to say that when, at least by analyzing the transcriptome in the EVs, I didn't see. I didn't look at properly, but I didn't see that it was like a huge change. But I also have to say that these EVs or this differentiation is mainly endoderm and mesoderm, and they also generate some neurons. But what I saw was like a massive defect also in Wnt, beta-cat, that is also regulating the gastrulation and mesoderm, endoderm specification. And regarding the retinoic acid, I don't remember I saw like something that at least in this transcriptomic data, it was not showing like a huge difference. Maybe if we sequence later on, because this was only 96 hours of EVs, I can extend until 9 days, and 9 days later, I saw neurons that are attaching into the plate. So maybe at that time, it's something that we can see.
I mean, I would be really surprised if you are blocking retinoic acid differentiation.
Yeah.
I have a question about, you started with a slide from Fiona Watt's work saying that a knockout of NF-kappa B has a phenotype in ES cell differentiation, right?
Well,
what did she exactly describe? What I'm driving at, do your new insights about I-kappa B explain her observations?
I have to say, it could be like that we cannot discard never that there's no any NF-kappa implication. But what they described was that when you overexpress NANOC, you block some of the expression of relay, RLB, CRL, and then when they measure NF-kappa activity and then try to stimulate the cells, when you overexpress NANOG, NF-kappa B activity was totally abolished. And when they, in the other way around, when they overexpress the rel, relay, relB, they induce a loss in silver renewal and also an inhibition of the pluripotency. NANOG and O4, it was totally reduced. But also very curiously, when they also use a kappa B alpha super repressor, which is you mutate the serine residues that are phosphorylated. So I-kappa B is phosphorylated in serine 32 and 36. So when you mutate these two residues, I-kappa B cannot be phosphorylated. So this creates a super repressor that is always binding NF-kappa B, and when you try to stimulate the pathway, since it cannot be phosphorylated, the pathway doesn't respond, and you always block the NF-kappa B activation. So they also introduced the kappa B alpha repressor, and they observed that they were getting like an increase in NANOG expression and pluripotency, let's say pluripotency phenotype. But they didn't go further. So this is something that they just described, like they were having more NANOG or higher NANOG expression and higher SEC1, but then they didn't go further.
But I'm still not entirely clear what they did because I didn't read these papers, but could it be that it has an effect on the levels of I-kappa B when they introduce these mutations or these perturbations? I mean, could this be an indirect effect on I-kappa B which would then relate to your work?
I think, I don't think so, because the main function of this, you remove I-kappa B and then can affect the NF-kappa B. But there are other I-kappa Bs that are very similar in terms of inhibiting the pathway, such as I-kappa B beta, and it has been demonstrated that when there's no I-kappa B alpha, I-kappa B beta can substitute the function of inhibiting NF-kappa B more or less in the same way. It only changes the speed in which the pathway is activated, but just maybe a question of minutes, not longer.
And why does this not happen in your work? The compensation with
I didn't see
the beta form?
When I checked all the genes from the NF-kappa B signature, like to see, targets and effector proteins, you see here that I didn't really see a change between wild type and knockouts. So you see that all the expression is almost the same between situations. It doesn't cause any effect, the lack of kappa B alpha in the NF-kappa B signature. So maybe, of course, I think we need to look, because also I think this is important to see whether without I-kappa B also they can respond to NF-kappa B activation. Of course, upon differentiation, because in the muscle cells I tried and I'm not obtaining much. But I think, that this could be explained because of other I-kappa Bs that are rescuing the function of NF-kappa B inhibition.
Luis, what's happening upstream? I mean, I know that this is a huge question, but at some point you were expecting this, no? So have you checked, what is your thoughts about in physiology, what's going on? What is activating I-kappa B, the nuclear function to maintain pluripotency? What's going on upstream? Have you measured IKK levels or?
AKK beta, I think it doesn't. It's not much. I think it doesn't have much to do with AKK beta. I think it could be. It's just a speculation, but I think it could be controlled by pluripotency network, because I found I don't remember the figure. It's in the figure. The figure 24. I identify a putative enhancer region for I-kappa B alpha gene, in which NANOX, was binding. So I target the region with a CRISPR-I approach to silence that region, and I saw that the gene of the expression of ECA-PAB alpha gene was totally reduced. So I think, of course you always need to check NF-kappa-B how it is, but I think maybe the control of the expression of the ECA-PAB alpha gene is more related at this stage with pluripotency. And also checking other data, like the programming data, and also from naive to iPSCs. The levels of ECA-PAB is when you go from naive to prime, they are reduced. And when you start to reprogram the cells, you start gaining ECA-PAB alpha. So maybe this could be a signal that the pluripotency network is controlling ECA-PAB alpha expression, which also makes sense when you look at the NF-kappa-B, because since it's an inhibitor, and you need to have an NF-kappa-B inhibited, if you are also controlling by the pluripotency, you are indirectly inhibiting NF-kappa-B signaling through ECA-PAB. So this, I think, it's a bit, a bit mess, but I think it's also another possibility. I think now with the mutants, I think it's much better if we just do knock-ins and see and measure all this transcription and control of ECA-PAB alpha and see what is happening. So.
Can I open the reprogramming melon now? No, because of course, it's one of the parts that I was really curious about, and when I was reading, and I was like, I want to know much more. I know that this is complex, but this is so interesting at the same time, given the role of NF-kappa-B in reprogramming in a cell intrinsic manner, and the phenotype that you are observing, that apparently there's an increase in reprogramming efficiency that could be, mediated by NF-kappa-B, the cytosolic function, the cell extrinsic function, and IL-6 and something else. But you have the nuclear function when pluripotency program is, starting. So what would you do next to explore this?
I think, of course, because we isolate MEFs from knockout embryos, so I think we observed a reduction in the efficiency, like half, 50% less, but the cells that were reprogramming were having the colony was bigger and all the colony was totally pluripotent. So I think that maybe I would just first checking how is the DNA methylation level, because unless in the methylation level, because this is one of the block routes of the reprogramming, the lysine 19 methylation. There are many papers that when you remove this mark, you increase the pluripotency. So maybe this could explain, maybe in part, I don't know, whether these higher or bigger colonies is you need maybe ECA-PAB in different phases of the reprogramming, because this is also another history. But maybe it's required for forming the colony, because we are observing that are lower number of colonies. But maybe to establish the pluripotency, or to stabilize the naive or a more naive program of reprogramming colonies, actually it's better if you remove ECA-PAB, because if you think in the other way around, maybe what we are seeing. One thing that I would like to do is take the IPS. I didn't have still time to do that. Take the IPS and try to differentiate and see whether after, once they are reprogrammed, try to differentiate or still they are blocked in the naive state. But I think it's also very, I'm not an expert in the reprogramming, honestly, but I think it's even more complex, because I think you need to check in every phase, analyze if it's required first, then remove ECA-PAB, or start maybe playing with some Digron, try to wash ECA-PAB from the beginning, or let it in the beginning, then remove it when you start to, you need to activate more endogenous pluripotency genes and see. So.
No, but also maybe it's a good way you have the mutants, that are able to bind to the chromatin, or with these you can dissect maybe which is the form needed for reprogramming or for the establishment of pluripotency and the maintenance of pluripotency, which is, I think, is really one of the results of your work that is really interesting.
Yeah, I think with the mutants now we can exploit like lot of things.
Or also working with inhibitors, with IL-6 inhibitors, or the shutting down the cell extrinsic pathway of NF-kappa B, just to dissect, this complex network that you are revealing in reprogramming.
I think this is crucial with now. By anything, I would make the mice to work with with the knockings and all this, and I think it would be first with a mice we would be able to know what is happening specifically with this chromatin function of kappa B alpha upon differentiation more in vivo, and also of course just with MEFs isolate and regarding the reprogramming.
If you could also test this of course this is because of my background, but I was speculating what's going on in vivo reprogramming. If you cross this with reprogrammable mice, so you would expect more reprogramming but less teratoma formation, right? Because knockout cells wouldn't be able to form teratomas because they are not able to differentiate. So you would have huge dysplasias and the differentiation, but not teratoma formation, right? And we haven't seen anything like that in the field so far, which is.
I have to say also that since the degree of the differentiation in all the tissue in vivo when there's no kappa B is much less, so they are less differentiated. Like if you take, for instance, some intestine and you see that the degree of differentiation is much lower, so it's not like as well defined as wild type. So I guess maybe that since if they are less differentiated in vivo, when you try to reprogram, maybe the efficiency is higher, but it's totally a speculation.
Very interesting. And maybe a more provocative question, because you are measuring these changes in cell identity, the differentiation, reprogramming. Do you think it's playing a role in differentiation or just changes in cell identity?
I think cell identity.
Cell identity.
I think which is because since we still need to define better the lysine 9 trimethylation landscape in the knockout. But I think this lysine 9 trimethylation is crucial to keep a good cell identity. And since we are also seeing some phenotype related at the end with cell identity in adult tissues or in formed tissues, this mark is also very important at that time, when you need a very stable silencing of other differentiation programs and pluripotency. So I think it could be a symptom of a problem of cell identity that also are causing a loss of DNA methylation and also other maybe printing problems that could be from this. This is the result from the lysine 9 was from 2 weeks ago, and so I think we still need to much better define everything, but I think with this, we are seeing now with the lysine 9, which is also crucial in more adult tissue, I think I would say that this is a cell identity problem.
That's a huge statement, right? Because
it's
so interesting.
I don't have any proof yet.
No, but it would be easy to test, with different conversions from fibroblasts to other cell types, cardiomyocytes and muscle cells, B2T. It is if it's controlling at the end the epigenetic program, and it's true that it's controlling cell identity changes. It could be interestingly expanded your results to a much broader field. No?
Would be amazing. Of course, it's just totally speculation because now I think there are many things to study and see and
I think you probably, to answer that question, you need a regulatable form, an inducible form. You have to be able to degrade it and restore it, and then you can really study that. I have another question. Are there any mutations of I-kappa B that are related to developmental diseases, or
There are mutations of IKB in some lymphomas, and Hodgkin lymphomas and B-cell lymphomas, and also in glioblastomas, especially in the ones that are having poor prognosis. So actually, regarding the glioblastomas, Dr. Luis Espinosa has started like a collaboration now to further study this. But it's been demonstrated that when you try to overexpress in glioblastoma cell lines IKB, then they stop growing and they die. So I think this is also important. I mean, there are groups working on that, and on a specific role of IKB alpha in cancer. So I don't know.
But they're not changing the distribution between the two modes that you showed, chromatin and.
I think this is something that now they are exploring, because now like try to define how much of the function of is depending on the chromatin, how much is depending on the NF-κB. There are some of the cancers are also related that have a mutation in NF-κB alpha. Some of them are having also a severe NF-κB activation. But I think now with specifically with this kind of mutants, I think it's possible to discriminate, because so far it's been very difficult. Because and so it's a protein that has a lot of ankylin repeats, so I think defining specifically the interactome in different situations I think is crucial. Even like now that we have the mutants, try to do mass spec to try to better define the complex in which IKB alpha could be maintaining at the pluripotency and also upon differentiation exit will be, I think, also very nice to see, and also to define like a more definitive or like a specific function.
Maybe also in cancer it could be possible that it's maintaining the stemless features in tumors, right? So those population more stem or more cancer stem cell like are also having higher levels of phospho-IKB.
This will be in line with what we are seeing now here with these results. I don't know if now people are studying that, I hope. But yeah, it would be good support what we are seeing here, that they are like more drawn towards a more or less differentiated type that are blocking the response to chemotherapy or in immunotherapy.
This is in line with the question I asked you before, if it's you think that IKB is maintaining pluripotency network or is just maintaining cell identity, right? Which it will have different implication in cancer as well. Important but different, no?
Yeah, I think, well, it's true that I don't think this is something specific of the pluripotency state, because then when you take some adult cells from some intestinal cell lines and you remove IKB alpha, then they cannot properly differentiate neither. Just with a cell line that normally is wild type and you deplete already in a cell line the IKB alpha. So I think that's why also I was thinking that maybe it could be also a problem of identity, because if already when at least the tissue is like more formed, then if you deplete already there and try to differentiate, it's having also problems in that differentiation. So it's maybe would suggest that could be also more related to cell identity. Yeah, but as you say, these are very big words, and it's just we don't have still any proof of that. So.
Something else? I mean, we have spent a lot of time discussing. Because we just this discussion and not in terms, I didn't have the opportunity to congratulate you. I congratulate your director, all the team, the family and friends that are out there, which are also very important during this important period of your career. I have enjoyed a lot the book, the thesis, and mainly the discussion. That I think it has been really, really rich, and you have been really comfortable addressing all the questions. And with that,
I also want to congratulate you, and also for a nicely written thesis.
Thank you very much.
And presentation.
Thank you very much.
Nobody wants to add anything else? Great. So then we will ask you to leave the room. for the lead.
Thank you.
think everybody's in the room already. Yes, Luis, it has been a pleasure for us to be with you today. And we all agree, the committee agreed to give you the maximum qualification we can give you so far. That is excellent. And someday you will receive the notification about the cum laude. So congratulations to you.
Me ha proporcionado, o sea, ha sido muy rico tener a vosotros y todo el feedback que tenéis. Yo lo agradezco mucho. Y a todos vosotros que, bueno, habéis estado aquí todo el tiempo ahí apoyándome y en la buena y la mala. Y nada, o sea, gracias a todos.
A ti, a ti.
y ahora tenemos pica pica allí en la Inner Square del Pierre Louis. Así que estos dos invitados. Y que os espero ver.
Enhorabuena.
Muchas gracias.